Medical Journal Research, Gene Editing by Sadie Laster. Gene editing refers to the practice of altering one's genetic material done with the aim of improving a characteristic or correcting a genetic disorder. This usually involves deleting a DNA sequence and or replacing or inserting a new one. This is very important because millions of people are affected by genetic disorders, cancer or HIV, and other disorders that are caused by mutations, and genetic editing can promise to cure or prevent these certain disorders. The basics of biology you should know are mainly about DNA. DNA is the coding information for all organisms, the blueprint of life. It is located in the nucleus of a cell and twisted into the double helix shape you can see on the right, and is made up of different base pairs, which are individual to the person as it is their genetic material. Also important are enzymes, which are proteins in organisms that produce specific biochemical reactions depending on their type. Also important is RNA. RNA is transcribed from DNA and is used as the message that goes throughout the body, specifically to ribosomes, which read it and make certain proteins that build the people we are. Specifically used in gene editing is the CRISPR-Cas9. This is a process found by scientists, but CRISPR itself means clustered, regularly interspaced, short palindromic repeats. This is quite a mouthful, but what it actually means is in cells we found sections where read from the 5' end on one strand from the 3' end to the other strand is the same from front to back. Basically, if read from the front from one side or the back from the other side, it is the same. Cas9 is also important. This is known as actually CRISPR-associated protein 9 and is known as molecular scissors by geneticists as it cuts the DNA. Basically, the CRISPR process was found in bacteria first, as bacteria used the CRISPR system as their adaptive immunity, protecting them from viruses. Essentially, bacteria that would use the CRISPR system to cut certain sections from the virus so it could recognize it. It would insert it into this CRISPR array section, the section of these repeats, and then in the future, the CRISPR could make RNA sequences that would pair with the Cas9 protein to recognize when the virus returns, and they would be able to efficiently destroy it with its cutting process. How we used it is scientists edited it to find a certain sequence of the human genome that causes the problem, basically the mutation in our base pairs that causes disorders. Then we create a guide RNA, which would be paired with the Cas9 protein and would cut the certain sequence we want. Then scientists, if they leave it cut, will basically just turn off the gene. It won't be able to be used anymore. So if there is a specific problem in that gene itself, it'll just be off. Even better, scientists can invent this template DNA, which is how they want the base pairs to go, and by inserting it back into the cell will want to repair in that certain way. So scientists can basically edit the DNA to repair itself how they see fit. Why we use the CRISPR-Cas9 is because it is fast, effective, precise, and cheap. It's not super cheap, but compared to the previous techniques we were using before, it is much cheaper. As well, it's very easily programmable, so that way it can be used for all living organisms for all sorts of reasons, and not just in animals or plants or just humans. Some challenges with the CRISPR itself are laws. Gene editing is banned or just has many restrictive rules in basically all parts of the world, so it's hard to know if we'll even be able to use it if it's banned everywhere. The cost as well, as I touched on earlier, it's quite expensive, so it's unknown if it'll just be able to be used for everyone or rich people, but so far it looks good, but who knows. Also, right now, we're more focusing on rare diseases and in animals, so people don't know when it'll actually get to diseases that affect humans commonly. Actual examples of gene editing in the real world is that Chinese scientists use CRISPR technology to edit twin girls before they're born to be immune to HIV. This is fascinating because gene editing was actually used in humans and these babies survived, so maybe gene editing could be used. As well, we use gene editing to disable the gene in someone's immune system that attacks the foreign cells, so that patient could take donated blood of a different type and it didn't attack these donors' blood cells. Also, by studying mice, we learned that we could cure their different genetic disorders, and we even slowed or reversed signs of aging in these mice. Some other interesting examples are China created micropigs to be sold as pets. As well, humans were able to invent these chicken eggs that will not trigger chicken egg allergies in children and just in general. And we made certain crops, such as maize and rice, resistant to bacteria. So overall, it's being used in all sorts of fields, medical fields, pharmaceutical fields, and just farming. So it shows that it has uses everywhere. Some positives of gene editing is it can help with prevent and treat diseases, as I touched on earlier, cure genetic disorders. Even we could alter humanity to be more intelligent. As well, we could edit plants to be more resilient and can produce more, so this could help end famine. As I touched on with the mice, we could start delaying or stopping aging. As well, with the research that would come along with human genome editing, we could see the changes in how they work. And so that way, even other disease treatments that aren't gene editing might cost less because we could find more efficient ways. Some scientists even add that due to the controversial nature of gene editing, more scientific communities will pop up because of the nature of this. Some negatives of this, however, are people worry that people are just going to start making designer humans, which will adhere to more traditional looks, which is basically eugenics, which is not good. As well, we don't know how future generations are going to respond to gene editing, which is why it's banned in many places, because we don't know if it's going to create a chain effect or how it's going to affect many generations down from us. Because right now we could be fine, but later it could be very bad. The other issue is consent, as this is mainly done on unborn children and embryos, so they don't really get to say yes or no, but they're still getting gene editing done to them. As well, if we keep doing gene editing, the more we do it, people say it could be more dangerous, because if you keep repairing or fixing, we're bound to keep making mistakes. As I touched on earlier, because of the cost, it could be available only to the higher class, which could lead to a wide class difference, because rich people will even have genetic advantages over poorer people. People worry the technology may just be used for enhancements for especially the rich, rather than actually helping with diseases and hunger. So far, gene editing is a very controversial topic. It looks like it has outlooks that could help humanity in general, but it also could be very dangerous. So people should tread lightly, but not disclose themselves to this option entirely. Thank you for listening. I hope you learned a little about this very interesting topic.